Next up, I'd like to welcome Dr. Brian Kennedy. I already spoke about his impact in this field. He's in Singapore for a number of years, leading the Healthy Longevity Center in Singapore, and is an academic that also does a lot of work with clinics and with businesses, and today is going to explore a topic of aging clocks. So please welcome Brian Kennedy.
Okay, I'll take that as a yes. I think I'm on. So I'm back again. If you were here yesterday, you're probably bored with me already. And I'm just warning you ahead of time that I'm going to say the word biologic aging clocks about 90 times in this 30 minutes. So that's going to be a lot of drinking for you guys. Really today I'm going to get more into the science. And I'm going to start with one slide that I used yesterday. And talking about interventions and biomarkers. And really my lab works a lot on interventions. We try to develop new small molecules that slow aging and test a lot of different interventional modalities. But without having biomarkers to measure age, how are we going to determine which of these interventions work? And it's true that these biomarkers need to be validated, and most of them aren't right now. But we have to work toward that as equally hard as we do toward developing the interventions and then bring them together to get the validation. And when it comes to measuring biologic age or coming up with an algorithm that estimates biologic age, there was a major breakthrough in the early 2010s. I would say that around 2010, the general consensus in the field is we just can't develop good biomarkers of biologic aging. There are individual markers that work quite well, that correlate with age, but they're not good enough to really measure biologic age or predict mortality. And there are some good measurements, the physiologic measurements and functional measurements in certain populations, like time get up and go and gait speed and things like that, that work well in people over the age of 70. But what we need is a life course measurement of how people are aging. If we're going to intervene early to 40-year-olds, for instance, we need to know some way to whether these interventions work or not. And so Hannum and Horvath came up with these biologic aging clocks, and I want to dig into that a little bit more and tell you what they really mean. Now, therefore, on the right there, what you'll see is you have a chronologic age, which your passport tells you, and you have a biologic age with these clocks attempt to measure. And so you might find somebody that's chronologically 50, but biologically much younger, so they're aging well. That would be the person on the bottom. Or you might find somebody that's chronologically 50, but they're biologically 60 or 65, and those are people that are not aging well. Now, DNA methylation, which is one of the epigenetic or epigenomic tests you can do, how your DNA is modified in cells, can be one measure to determine aging. And some people have argued that it is sort of primal, that measuring how epigenetic changes happen into your DNA is a driving event in aging. And I think partly that's true. Certainly you can modify the epigenome and affect aging. But it turns out you can measure age from a whole range of different complex data sets. Basically, if you have a complex data set, and enough individuals of different ages, you can build a clock that predicts age. And so the first ones, and most of them still are based on DNA methylation, but they don't have to be. Now, what do these clocks do? What are they good at and what are they not so good at? We know at a population level, and I'm going to show you some population data in a minute, that these clocks work because the things that we know or believe we know affect the aging process also affect the clocks. So if you're physically active or you have a very healthy diet, in most of these clocks you're biologically younger. If you're male, you're biologically older. Men age faster than women. If you smoke, if you have heavy alcohol consumption, you're biologically older. Moderate alcohol consumption would be a long debate that we don't have time for today. Menopause accelerates biologic aging. So these are the kinds of things that we already thought we knew, and the clocks support that generally. Now, I need to dig in a little bit here because there are multiple types of clocks. There are first generation clocks, like the first Horvath and Hannum clocks and many others, that try to predict your chronologic age And then the residual error from that prediction is considered your biologic age. So again, if you're 50 and it says you're 47, the argument is you're aging well. But these clocks are a bit complicated because I'm not sure we know exactly what we're measuring. And in reality, that 3-year difference is a combination of the error in the algorithm and potentially the different aging that you see that's making someone appear to be younger. So those clocks have been very useful. They're still useful, but they're a little bit confusing. The second generation clocks try to predict an outcome like mortality or some set of clinical parameters that are associated with aging. And I'm going to make the argument today that these second generation clocks are more useful. And I'm going to tell you about a second generation clock at the end that we've generated. There are other kinds of clocks. So there's a pace of aging clock, like the Dunedin clock. So you can think of it this way. Most of the clocks are measuring the odometer in the car. The Dunedin clock is trying to measure the speedometer in the car. How fast are you aging at the moment? And I think there's some interesting data with this clock. We use it a lot in the laboratory. There are also organ-specific aging clocks. You can measure your liver age or your lung age. And there's a lot of debate as to whether breaking things down and measuring specific organs is more meaningful than getting a systemic age. No one knows the answer to this. I'm still on the side of measuring a systemic age is valuable because I think you're measuring that homeostatic network that keeps you healthy that I talked about yesterday. And then you can break that age down into components and let the algorithm select different subgroups. And I'll show you what I mean by that. So let's go look at some methylation clocks in population data. So I'm going back to Singapore. This is 6 million people in Singapore, 74% Chinese, 13% Malay, 9% Indian. And if you look on the right, this project we're kind of piggybacking on. This is a genomics project. And the reason Singapore did this is because together those three groups are like 60% of the global population. But in reality, they only represent 11% of the genomes that have been sequenced. So coming back to what I said yesterday, these ethnicities are highly underrepresented in biomedical data. So Singapore started this project where they sequenced 10,000 Singaporeans. They also did a little bit of molecular phenotyping and metabolic phenotyping. And we were lucky enough to get the methylation data and look at biologic age from this. Now, this is not a perfect cohort because the first thing they did is they brought a bunch of cohorts that already existed together. So this is not how you would ideally design a cohort. But it was a good start. In reality, however, they're moving way beyond that. So there have already been 100,000 genomes sequenced in Singapore. And pretty soon there'll be up to 1 million genome sequenced, which is about 20% of the population. So we'll get more and more data as we go forward. But I want to show you the data on the SG10K. And so at this point, we looked at 4 different clocks, Horvath, the Grimm age and Pheno age, and the Zhang clock. And they all somewhat worked in these populations, but not as well as they work in Caucasian populations. With the exception of Zhang, these clocks were largely made on Caucasian populations, and so it's an assumption that they're going to work in different ethnicities. The answer is generally they work, but they're not perfect. They correlate with each other, but not perfectly. That's what we see with clocks all over the place. And so this is kind of what we expected. We saw generally the same results among the different cohorts that were put together to make SG10K. And there were several conclusions we could make from this data. First of all, the Chinese population in Singapore is biologically younger by all 4 clocks than the Malay and Indian populations. This is not terribly surprising because aging often links to socioeconomic status, and the Chinese population has a higher socioeconomic status than the other 2 groups in Singapore. And so not terribly surprising, but interesting. The one on the right is also an expected result, I would say. If you have suboptimal metabolic health, you have an accelerated biologic age by a whole range of these different clocks and different measurements. The one in the middle is very interesting, and we're trying to think about what it might mean and how to interpret it. We happen to have a birth cohort, so this is methylation data on people right after birth and also their mothers. We could already see a difference between biologic age in males and females at birth. So if this is to be believed, baby boys are already biologically older than baby girls. And that means that something's happening in the womb, maybe genetic conflict or something else, that's already triggering the beginnings of an aging process. And I think most of us believe that aging doesn't start when you're 60 or 40, but much earlier in life. This would suggest it's starting prior to birth if this data were accurate. So we could do genetic analysis, so we could find genetic variants that are associated with accelerated epigenetic age, and we uncovered a number of these things. If you're an expert in the field, you might be interested in Wnt3A and PARP, which are two genes that have been linked to the aging process. Some of these other genes are interesting as well. I'm not going to go through this. Unfortunately, we only could validate these in the Chinese population because we didn't have enough data from the Indian and Malay populations. And this is one point I want to make. We really want to compare people in Singapore to similar ethnicities in other locations. So in that case, comparing, for instance, the Indian population in Singapore to the Indian population in Mauritius would be a really interesting set of data to accumulate. The takeaways from this study are that the Chinese adults experience slower epigenetic age acceleration. Males seem to be biologically older even at birth. This is linked to metabolic phenotypes. And surprisingly, a large number of these methylation sites that we're measuring are linked not just to aging, but to sex. And this is a point that I want to make. I think there are fundamental differences that we still don't understand between how men age and how women age. We can see this in animals. Many of the interventions we test work better in males or females, depending on the intervention. We know some differences like menopause, but I think we really don't have a good grasp on the differences in how men and women age, and we need to focus on this a little bit more. I think it also suggests that we need to think about ethnicity and gender-specific clocks, because these clocks, I think, will generally work, but they're not optimized for a particular ethnicity and maybe even geography, then we're not getting all the information we could from that. So if you look at clinical approaches to interventions, this is a major challenge in the field. How are we going to do an intervention and see whether it slows human aging? Now, the gold standard, and Matt showed you a little bit of data from this, the gold standard in animal models, of preclinical, non-human animal models, is to look at survival. Because, and I would still argue this is true, the one metric that, if you're only going to use one, that best captures aging is how long an animal lives. But this is not feasible to do in humans. So we have to find another strategy to measure aging. So there have been a lot of, I think the field's still grappling with this, and there's a lot of potential ways to do it. One is to do multi-disease prevention. So you take a large number of people and maybe 60 years of age, you give them a drug, and there's been a proposal to do this with metformin, for instance, a diabetes drug, and you then wait 3 or 4 years and see if they have a reduced rate of getting many different kinds of chronic diseases at the same time. Because as Matt showed you, if you slow aging, you should protect against many chronic diseases. This is a very expensive study. And in the metformin case, it's tens of millions of dollars. And metformin is a drug that costs 4 cents a day, so pharmaceuticals aren't going to pay for it. Pharmaceutical companies are not going to pay for it. And I really think that it's a challenge, because even if we could afford to do that with metformin, we need to test tens of different drugs and figure out which ones work best in which people and how to put them together. Biotech companies tend to target aging pathways, but then try to treat diseases of aging. And that makes sense, because they need to be reimbursed for whatever product they develop, and their investors aren't going to invest if they don't have some strategy to go to market and make a lot of money. But it's not really what we want to do if we want to understand aging. And so we've really focused on clinical trials using surrogate endpoints, these biologic aging clocks. And I'm going to talk about that a little bit more at the end, but I want to also mention that there's a lot of suboptimal data out there. Diagnostic companies are collecting data, clinics are collecting data. And one thing we really want is people to share data with us, because we're very good at analyzing data, not just my lab, but many labs in the field. And this data, even though it's not placebo-controlled, double-blinded trials necessarily, it still can be valuable to look at. And I'm going to show you a piece of this data just to give you an idea of what the strengths and weaknesses are of suboptimal data. And so this is a methylation company. I'm not involved with this company. They put on LinkedIn last year a data set that they had with 5,000 individuals doing a relatively simple methylation test that they developed. And this methylation test is you send saliva into the company, and they measure a very small number of methylation sites and estimate your biologic age. So it's a first-generation clock. But it's very rare to get 5,000 data points in a healthy population to look at. And these people are very interesting because they're very motivated to slow their age. So they're taking 1 to 4 supplements or drugs a day. These are very active people taking supplements. The clock is reasonably well. The mean error is about 3.8 years. And altogether, there were 900 different compounds that people reported taking, which to me is shocking because I can't name 900 different compounds that might affect longevity. I would get stuck probably at 100. And so people are taking all kinds of different things. And so this data was not really analyzed statistically ideally, but it did show that a product that we helped develop, Rejuvent, that we've done work on first at the Buck Institute and then later in Singapore, was the top of the list in terms of reducing biologic age. So I was very curious about this data, and I admit I was a little bit biased. If this data showed something else was working, I might not have gotten so interested. But I called the company and I said, can you send us the raw data? We want to look at what the raw data says and really analyze this data in detail. And Rejuvent is a product that contains alpha-ketoglutarate. This is mouse data, but what it shows on the right is that there's a 50% reduction in frailty when you give mice this supplement. It's a standard TCA cycle component that your body makes, but it makes less of it as you get older, and if you supplement that up, at least in mice, it has a big effect on frailty. So we got a graduate student, Camille Pabas, to really look at this data, and I've compared the Trumi cohort to the NHANES cohort, which I'm going to come back to later. This is a long-standing longitudinal study in the US, and I just think it's worth comparing a little bit. So there were 5,000 people in the Trumi cohort, and the first thing you realize when you look at data like this is it's messy. People are answering questionnaires about their age and their health status, and people lie. Even in questionnaires. For instance, 12 people reported that they were older than 120 years of age, which would make them the 12 longest people ever on Earth. It's highly unlikely. So you have to have legitimate, unbiased strategies to optimize this data set to get it as legitimate as possible. Now, these people are healthy. Their mean biologic age was 2 years younger than their median actual chronologic age. And you can see that in the data. They don't smoke. They report excellent health. They're more males than females. Males are more interested in these supplements these days. And like 2/3 of them, or more than 2/3 of them, use supplements. So we had a lot of data to analyze. That's the good part of this. And the clock looks like other biologic aging clocks. So the red line is chronologic age on the left. The blue line is biologic age. And one of the things you'll notice as you get older using this clock, people are biologically younger than their chronologic age. We know at least one reason for this, because if you're 80 years old and you're not aging well, you're probably not sending your saliva in to a company to get your biologic age measured. You probably are not alive to do that. There's something called survivorship bias. So the people that are older are likely to be biologically at or younger than their chronologic age. There may be other factors engaged in this, and we've tried to regress that out as we looked at the data. The traditional risk factors work with this clock. Smoking's bad. Waking up rested like I did not do today is good. Being in excellent health, people know pretty much where they are. So the average person is 2 years younger. If they reported excellent health by this clock, they're 3.3 years younger. If they reported poor health, they're 1.9 years older. And so then we started to go in and look at the actual supplements. And everything with an asterisk is significant. You'll note that there's a very intense all data and also a non-user data over there. So these are the whole data set. Again, the average is 2 years younger. Most people taking supplements either had no effect or it had a negative effect or a positive effect on their biologic age, meaning it reduced their biologic age. Rejuven, which I list here, which I am involved with, full disclosure, it's a time-release alpha-ketoglutarate plus low-dose vitamin A if you're a man and low-dose vitamin D3 if you're a woman, was associated with a 5.7-year reduction in biologic age, again by this clock. Anybody taking AKG also had a reduced biologic age. There are many other brands of AKG, but there was a bigger effect with Rejuven, maybe because of the sustained release form. Other things worked as well. CoQ10 was associated with lower biologic age, and curcumin was associated with lower biologic age. I want to focus on this thing in the middle with the asterisk NAD boosters. It only had a 0.5-year reduction in biologic age, but we had a lot of data points, so that turned out to be significant. And that's a small effect, but I think this is one of the problems with this kind of data set. So about a year ago, one of the NAD precursors is NMN. And we don't know, when people say they're taking an NAD booster, we don't know what brand, where they're getting the NMN or the nicotinamide riboside from. It's just a compilation of people taking it from different sources. So on the left, what you'll see is people that we bought, and academically, but we didn't tell them we were a university, we brought 16 brands of NMN from commercial sources and tested how much NMN was in the NMN. And if you look at the dark red there, those are 3 sources where there was 0 NMN in the product. And all of the stuff in forms of red and pink, there was a reduction in NMN relative to what's reported on the label. So that's one of the problems with the supplement industry is the quality of the supplements is highly variable. Some of them are great, some of them are terrible, and the consumer has no idea where to buy their supplements from. Some of these supplements cost up to, the NMN supplements cost up to $3,000 a month. So people are spending a lot of money on, in some cases, stuff that is not what it's supposed to be. So when you look at a 0.5-year reduction in biologic age, that's a compilation of people that are taking things that don't work and don't exist and are NMN that doesn't exist in the product. Some people are taking the right thing, etc. I need to move quickly because I want to get to the last part of this. In the Rejuven case, we found older people do have a bigger effect, and males have a bigger effect than females, and we're still looking at this. So if you look at the story right now in the aging field, we've made a lot of progress. We have interventions, we have diagnostics, clocks, we have pathways, hallmarks, and we have outcomes, diseases, and functional decline. And at least what we need to do is connect these verticals together. And at least for this data, we can say that Rejuven has a big effect on the TrueMe clock. Now, if we looked at another supplement, we might find a much bigger effect on some other clock. And so I think we need to do more of this, connecting these verticals together. So quickly, you don't have to use methylation. And I agree with a lot of the problems Matt brought up with regard to consumer use of methylation clocks. There's a lot of noise in the data still. I think a lot of people are working hard to fix that, and I think there's a lot of good science, a lot of good effort going into that, but there's still a lot of variability. And also the problem is if you go to the doctor's office and say, there's 13 methylation sites that tell me I'm too old, the doctor's like, what am I supposed to do with that? How do I move those methylation sites? It's not really actionable. And clinics are scared of that because if they measure the methylation sites and give you an age and then give you a set of interventions to do, they don't know for sure that that methylation is going to change when the person comes back 6 months later. So we decided, what can we use that hospitals are going to get and understand and that's really reliable data? And to us, that was clinical parameters. HbA1c is measured more or less the same in clinical labs throughout the world. The data is very accurate compared to other measurements. And so if we use measures like that, it's very reproducible. So Jan Gruber and Feng Sheng in our program at Healthy in US use this enhanced data I mentioned earlier. There's multiple years of input. So they use 1 year of input to build a clock, and this is a second generation clock. So it's not really a biologic age, it's a mortality risk assessment. So if I tell you using this test you're 50, that means you have the mortality risk of a 50-year-old, assuming the clock's working. And so they built it on 1 year, and you can see the data here. You got this dark blue box. Those are people that have a high mortality risk relative to their chronologic age, and the turquoise box are people that have a very low mortality risk. Those are people that theoretically are aging well. And so you could test it on the next year of input from this study. And what you see, and again in the turquoise and dark blue, is that the quartile of people that we scored as the youngest have very low mortality over 200 months compared to controls, which are in black. And the dark blue, which are the people that have a higher biologic age in our assessment, have a much higher mortality rate. So this is doing a very good job. In fact, we think it's doing the best job of predicting mortality. There may be other things out there we don't know about. I'm going to go through this very quickly, but we could also break this NHANES data down into different groups. And so if you look at that dark blue line, those are people that are optimal health in terms of these clinical markers. That means every clinical marker that was analyzed is in a reference range that we call normal. And that declines as you get older, as you might expect. It's much harder to keep people clinically optimal. Those people have a lower biologic age, of course, by our test, and they have much lower mortality. You can see the mortality curve on the right and optimal. But you can break that group down into two subgroups. One subgroup are optimal, no drugs. So this is 60-year-olds we're looking at here, 65 to 75. So if you go to the doctor's office and you're at 65 to 75 and all your clinical markers are in a range, the doctor's generally going to tell you, hey, for your age, you're doing great. Just keep doing what you're doing. If you can walk a little bit more, do that. And those people are biologically younger, but what we found that's interesting is the people that were taking medication, we knew what medications they were taking, and were clinically optimal, had a lower biologic age and lower mortality than the people that we were calling optimal. And I think this is an argument that we need to be acting earlier. It's not good enough to say you're generally doing okay. We need to be doing healthcare throughout the life course and intervening as early as possible to try to keep people healthy. It doesn't have to be drugs, it can be lifestyle, but we need to be optimizing these clinical as much as possible. Now, just briefly, our clock, the clinical chemistry clock, lineage2, outperforms the methylation clocks. So if you look at the first generation methylation clocks, at least by NHANES data, they don't predict mortality at all. Now, they weren't designed to predict mortality, but you think if they're measuring something relevant to aging, it should be linked to mortality. The second generation clocks do pretty well. In fact, GrimH2 does quite well. We're statistically better, or we have a trend toward being better with all these clocks using the clinical markers. And it's better at predicting cognitive decline and gait speed decline as well. But the question is, can we use it personally? And we have data on 100 people now so far moving forward. And I just want to show you one of them. This is a clinic in Singapore that really is focused on metabolic health, not aging, with Suen To. And I just want to we picked one person out, and we wanted to pick a person that was already doing well. It's easy to say somebody that's biologically really old and doing poorly, we can improve. That's not that surprising. But this person came in, they're 62 years old, pre-diabetes, a little bit high BMI, but 62, they're pretty healthy. And in fact, the lineage clock assessed them at the mortality risk of a 54-year-old. But what we do is we don't just give them that number. We break down all the principal components. So we're using a linear approach that identifies principal components that each go into this age prediction. And so each of those PCs is contributing to the lineage prediction, but each of those PCs is composed of different clinical markers. And so this person was put on, I'm getting there, Martin. So give me 2 or 3 more minutes. I just don't want any questions because I'm scared Matt's going to ask me a question, so I'm using up all the time. And so the principal component 1, even though this person was doing well, is accelerating their age by 4 years. So we thought, what can we do about principal component 1, which is, if you look at it, is mostly metabolic markers. So this person was put on lifestyle changes for 3 months and a GLP-1 agonist for 1 month. So after 4 months, it was measured again. And you can see this principal component 1 is brought down dramatically. And this person went from 8.5 years younger mortality risk to 13 years younger. But the point I want to make is the other principal components went up. So that you can see over here, this one went down, but this one went up. And so now the next iteration is to take this person and target that principal component and see if we can get to sort of a minimum mortality risk for each individual. Now you'll notice this is personalized and precision because each principal component is composed of different clinical markers, pretty much all of which are actionable. So we think this is an actionable clock that's getting us toward personalization and optimizing age. So I'll go through this quick. This clock identifies existing disease. It shouldn't have to, but amazingly, a third of people that come to longevity clinics have existing disease they don't know about. But more importantly, identifies subclinical disease, and it seems to work on a personalized level. So we're developing this clock now, both academically, happy to collaborate, and also commercially with longevity clinics, hospitals, and other people interested in doing this right now. So this is in progress. We'll have a lot more data soon. And one of the groups that's helping us do this is Martin, Odie, and Alice, who is incorporating our clock into their app. And so we're excited to work with different companies and different strategies, but Alice is one that we really are excited about, and they're in the process of raising money. We have a consulting company also that's separate, and there's a lot more information on there if you're interested in getting more info or just interacting with us. And thank you, and sorry I went over, Martin.